Hello again. So yes, as Randy mentioned, we thought it would be a good opportunity having sort of looked under the hood, so to speak, and got a baseline familiarity with the layout of the database to launch into some more specific queries and really explore the results a little bit deeper. So that's what I'm going to do with you, walk you through some example queries. So again, these queries will be primarily launched from the home page. One thing I didn't point out earlier that I'd like to mention is that it's not shown on this particular screenshot, but if you are online, in the bottom corner here, you'll see a date. The IDB was last updated, and we refresh on a weekly basis. And I mentioned to you, we are basically keeping up with new publications in PubMed. And so if you are querying the database and you come back and revisit it, you can tell if the data are the same or different based on that last update that you'll see at the bottom corner there. All of the exports also are time-stamped so that you can easily refer back to versions. All right, so I'll begin by with the specialized search drop-down menu Randy had mentioned. From the home page, you can access alternative search types. So the first four termed detail searches, we're not going to go into detail here with those. They're available to enable you to search any and every field in the database. So that's really, the like I said, the field level bread and butter of the IDB. But as the remainder of the search interfaces have really been optimized to allow for vast majority of searches, we're going to focus on those. If you like to use them, or if you have specific questions about them, of course in the breakouts we're there to help you with those. So what I would like to look at in a little bit more detail is the so-called identifier search, which was, you saw this slide earlier. So every, whether it be an epitope, a particular submission, assays, etc., are automatically assigned IDs upon input into the database. And those IDs are static, so you can always use them to come back and retrieve very quickly, for example, data from a particular reference, by putting in that PubMed ID, if it's your own or someone else's, and quickly getting your results that are, of course, filtered exclusively to data in that reference. Everything else is as you've seen earlier, except that it's specific to one and only one reference. There is, you just also saw our 3D browser. So this internal tool that allows you to visualize when there has been a 3D structure involving an epitope curated. So there's another way to get at these by a browse function that's available from this menu. And this browse is organized as a tree that has three high-level branches: B cell, T cell, and MHC ligand structure. So at the highest level, these represent, you can see here, the source of the organism that's essentially the receptor, the immune receptor. So you might be interested in an epitope that binds to an antibody. You could browse the B cell branch. And in my example, I've opened the T cell structure. And essentially, if you're interested in a particular allele, for example, that might be involved in binding an epitope and the 3D structure of that interaction, you could essentially just keep, digging deeper into the various branches of the tree until you reach an endpoint where you'll see a count for the number of assays. And by clicking that count, you will actually then go back to the common results page with the tabs, the familiar tabs that we've seen. And as Randy indicated, this icon, this 3D icon, alerts you that that particular assay is associated with a 3D structure. And the way to launch the browser, or the 3D viewer, is to click that icon. So for this particular assay, you can see that 3D interaction by clicking there. And this is, again, the 3D viewer that you saw earlier. I'll just also point out that this is very customizable in the sense that you can define which parameters of the interaction you want to show. You can customize the colors. So, for example, show the one or all the TCR chains, show certain aspects of the MHC or hide them. And, you can manipulate it as such. So back to the home page, search interface. This is what I'll use as kind of the launching point for the first example query. So in the epitope panel, there are, you saw earlier, various ways to define in terms of linear epitope the level of stringency that you place on the results. So, So in this particular case, I'm doing an exact match query for the this is the very commonly well-characterized flu nucleoprotein peptide. So if you leave this drop-down menu as exact matches, you will in fact get, results that are exact matches. As Randy mentioned, some cases we do capture peptides that might have modifications, and you can see the last couple records are in fact modified versions of that peptide, and you get your summary as well here by clicking the details for any particular record. In this case, if I ran the same search for the same peptide but adjusted it at a 70% homology blast, your results will then reflect, a broader array of results. In this case, we have exact matches, I believe one exact match at the top, but you immediately see single or double amino acid variants. These are naturally occurring variants. We know that because we can see sources associated with them in terms of the antigen and organism. So, these might be isolates or what have you that were studied, or peptides that were made with known mutants and studied. Below, however, you see a row where there's a peptide with no natural source. So this is an example of what you could call an analog in the database, where there was, for example, an artificial alanine mutation introduced into the peptide to study the importance of a residue, for example. And that will be captured in the database. But as we've loosened the stringency, we will return analogs as well. So we often a question that comes up is, if I have an antigen or a sequence of interest, how do I predict epitopes from that? And we sort of jokingly say, well, first thing is you don't. You query it, look for experimentally validated data, and then, of course, we encourage you to use the tools as well. But it's the first stop can always be look for what's out there, especially if you're not completely, familiar with the entire body of literature for that particular sequence. So in this case, we could still utilize the search, the linear search query, by inputting in a much longer string. In this case, it's an entire protein that you don't see the entire sequence, but hundreds of amino acids. And I've changed the constraints to substring. So the substring now returns peptides that are matches within that longer stretch. And so you can see here, each of these is a hit within my longer protein that's displayed inside of my filters. And, this is like essentially a way of studying what's known from your protein, right? In one sense. So utilizing the homepage search in this example query, we had kind of we've shown a couple examples now, but now you haven't seen the finders and the utility of them. You can perhaps feel a little bit more comfortable, hopefully, defining and utilizing each of these tiles. But let's, for example, suppose I wanted to find Plasmodium falciparum class 1 restricted T-cell epitopes defined in humans. Every narrative of a query that you think of in your mind, or every sort of sentence, can really be dissected down into some of its query parts. So that's what I've done in this case, just kind of box the relevant parameters with an arrow to indicate where each of those will go. So of course our source, in this case at the organismal level, will be Plasmodium falciparum. Since we're, in this case, it's not allele specific, it's somewhat broad, but we can define at the class level with just radio button here. Since we're interested in T cell epitopes, again, we can unselect and choose T cell only. And then finally, the host simply can be defined by humans. The kind of more common hosts we have as radio buttons, but in the Finder, you can search any host from the NCBI taxonomy. So if you were to go through and input these, you would end up with populated search panels that would look as such. And executing the search by clicking the search, again, gets you back to the familiar sort of results page. So what I'd like to do that we haven't done yet is explore in a little bit more detail the assay details. So we've mentioned a couple times this morning that you can click on these IDs to uncover deeper information, more field level, and at the assay results, that's in fact what you're going to see. So this will be on a continuous page when you click on it. For clarity and the size, I've broken it up. So these arrows represent, you've scrolled down, but this is all collected on one page. So when you click on an assay details, the first thing you'll see is a summary of the reference. Again, the source of this information. And then just below that are clusters of related fields. So the data are categorized in groups that are interrelated. The first one being the information that's pertinent to the epitope itself as it pertains to its sequence or structure, its source, the position. This information may or may not be the same in terms of the positions as what's reported in the reference. So if there are some discrepancies, you can, for example, one epitope might be found at position 10 to 20 in one version of the protein or one sequence, and it might be shifted 11 to 21 in another. And that's, in essence, we look for it to be conserved in the source that we cite, but those positions might vary with what you see in the paper. So for clarity, we point that out. Also, you can always get a location in the reference, a data location in the reference. So if you ever wanted to go back to the manuscript and see the actual data, you could refer to the table 1 in that particular reference. That's where the curator extracted this information. So below that, we start to get into the what I referred to as the contextual information earlier. So this is the assay information that was used to characterize the reactivity of that epitope. So it's sort of flows in a nice narrative in that we would think of an experiment as beginning with a host, maybe some sort of exposure, immunization, and then you take cells or antibodies and expose them in vivo or in vitro and get a readout. And this flow kind of matches that experimental narrative. So what you'll see at the top is in the immunization related fields, what was the host. Below that, if there are additional details available, such as where the geography of the host is, where they were, for example, where the donors came from, their age range, in this case, very young Kenyan children. The first in vivo process, so this starts to get into what Randy referred to as jargon. So these are terms that we hope will be self-explanatory, but in not all cases, they might be a little bit, take some getting used to. So the first in vivo process describes essentially what was the biological agent that elicited the immune response. So in this particular case, you can see that it's described, the process type is described as an occurrence of infectious disease. So this is simply to say that it was a natural disease occurrence. So these are, we have drop-down lists that you can use to narrow those down. Below that, we see that if there is a specific disease associated with the host, in this case, they're infected with Plasmodium falciparum, they have malaria, and if there's a stage that's known, in this case acute, we will capture that information as well. The first immunogen is the biological agent that elicited the response. So since this is a natural infection, the immunogen will be the bug, the organism. So we use terminology to refer to the immunogen by describing its relationship with the epitope. So if the immunogen is the organism from which the epitope is derived, then the immunogen will be referred to as the source organism. So source organism means the organism that is the source of this epitope. And below that, of course, it repeats Plasmodium falciparum. In certain cases, you'll see a reference name. Again, this refers to what the reference describes the immunogen as. So details such as these were actually Plasmodium sporozoites. You know, in the NCBI taxonomy, that nuance may not necessarily be available, but it might be interesting to know that level of detail we could capture in like a free text field, right? Below that, there's another free text field called immunization comments. These are annotated by the curator, so they will input details to further enrich kind of what was happening. In this case, you're just told that there were 107 malaria-infected children. And then as we get into more of the assay aspects of it, you start to see the assay fields. So the T-cell assay fields simply describe the actual readout, the assay that was performed. In this case, it's a proliferation. What were the effector cells? So we have not only the tissue they were taken from, but the, if known, the phenotype of the effector cells. So CD8 T cells. Very interestingly, we also capture effector cell culture conditions. And what that means is how, if at all, were the effector cells treated before this assay? So were they re-stimulated? Were they taken directly ex vivo and tested? Is it a cell line that was created before assay? And so these are direct, but you might see examples that describe the culture condition of the T cell as a cell line or a clone. If there are antigen-presenting cells in the assay that, of course, are on the other side being recognized, presenting the epitope, those will also be captured. Similarly, tissue and phenotypes. MHC allele simply refers to the restriction if known or predicted. So we will capture that information here. Finally, on the readout side, the antigen, what we call the antigen is sort of, you could think of it as the biological counterpoint to the immunogen. Not in this case what elicited the response in the host, but what triggered the readout, either in vivo or in vitro. So for this particular proliferation assay, the epitope itself was used to stimulate the cells to proliferate. So we would call the antigen the epitope. Again, there's a relationship terminology that's used there. For example, if the readout was triggered by Plasmodium falciparum, the relationship between the antigen and the epitope would say source organism. But again, the details about the sequence, the biological entity, are always below the relationship as well. And then again, there is a spot for more comments by the curator. In this case, very often you'll see it as just a summary of the assay. So this is what I basically made into a narrative by going through these fields is more or less encapsulated by this, statement that was put in by the curator. To the far right of all of these detail pages, you will see links, and these are, as Randy described earlier, the interoperability that we've created, and these are various ontologies or sort of the authority in the field related to HLA, for example. Those are quick links that you can use to access further, deeper information about that particular parameter in the database. So, supposing I reached this point and I wanted to tweak the results, refine the query in some way. As I mentioned earlier, the search fields are always available on the results page to do so. So I could do something as simple as changing hosts. So it's currently set to humans. You could change it with a simple click to mice. Refresh the page by clicking search. You will need to click the search. It doesn't automatically refresh for reasons that Randy got into earlier. But this will turn green and you'll click search. It will also update your filters so that you can always confirm that it was correctly input. And then after doing that, all tabs will update. So you can see quite a few, back to the original query in humans, you can see quite a few results, quite a few assays, 146. Supposing I had a secondary follow-up question that was more about refining these results according to some sort of assay. For example, I'm interested in creating a tetramer and doing some sort of detection in my laboratory. I want to know, are there any known tetramers in this particular system? So we could simply, keeping all other parameters the same, come back and refine the assay from proliferation to tetramer. Again, in this case, I've just done it by doing the free text entry. You could also launch the finder and look at the assay tree and sort of use a single assay or a group of assays from the tree. But again, you'll refine the search, the results by clicking search. In this case, we have three particular epitopes that have been shown to work as tetramer in tetramer assays for 3 different alleles. So as an example, how you can play with the results, customize them even further. So you saw earlier maybe the power, Randy began to show you the powers that lie in exporting the results and working with them locally. So if I were to export these results, for example, I might further delimit this in terms of other parameters that I might be interested in. For example, in this particular case, I could filter on specific alleles. And again, this way you have all the data already locally. You can do filters, undo them, and you're not necessarily working through the search interface. The assay antigen. Let's suppose that I'm only interested in knowing positive responses when there was an epitope immunization, but when the response was triggered by the whole organism or an antigen, for example. Then you could, based on the assay antigen and the relationship I mentioned before, you could see most of these that are shown here were readouts done with, basically peptide on peptide or source organism on peptide. But you could filter out those that are at the source antigen level or broader source organism level. So this is a thing that we download and manipulation we do quite often. You might have seen earlier that there are a series of meta-analyses posted on our site that are really nice examples of how the database can be leveraged to do analysis of the known body of literature in a specific field, identify knowledge gaps. We call those meta-analyses, and one way that you could do that on your own side to identify areas of research pursuit would be to download the results and query them in these exports. Okay, so lastly, finally, I would as we've heard this morning and seen based on your feedback to us in advance, thank you for providing that, by the way, your research interest. It was very helpful to us to know in advance, what your area of expertise and interests are. We saw quite a few that are autoimmune, areas of autoimmune interest, so I thought I would show an example query for searching autoimmunity data. In this particular search, it's diabetes focused, but these themes, remember, can be quite easily applied across autoimmune diseases. So if you're interested in epitopes from auto-antigens in patients that are known to be suffering from an autoimmune condition, in this case diagnosed with type 1 diabetes, that can very easily be done by defining an antigen, a host, and a disease. So antigen of choice is up to you, of course. In this case, I took the common GAD antigen for diabetics. Humans will be my host, and disease I'll define as diabetes. So what I've done differently in this example is I'm actually running this query from the results page, just to kind of get you comfortable that you can do it in both sides, right? But remember, all of these query fields are available on the results page. And in fact, if you click search on the home page, having defined no other parameters, you're going to get all the data in the database, or more or less all the data that's predefined in those homepage search fields. And then you can take it from there and filter. So in my organism, I've defined it as humans. Again, you feel free to use the free text field or the finders in these cases. The GAD is also defined here. As Randy showed, I could go back and sort of stack antigens, so I could input multiple ones, and this number in green would update to reflect, and then you can also remove them if you'd like. And then so on the disease, free text search for diabetes reveals several different possibilities. You choose the appropriate one. In this case, insulin-dependent diabetes, whether your interest is type 1, type 2, may vary. And then back to familiar grounds, right? Back to the research results. And then, you get that information that might trigger ideas for further refinement. And it kind of becomes a nice iterative process of querying. So I think we being ahead of time, we have time for more questions. If you have some, please, Jay.
I have several questions, maybe in the afternoon.
Yeah, absolutely. We're happy to work with you specifically in the afternoon as well.
To get started, the first query was interesting. Plasmodium, what you showed. So when it is giving a spreadsheet containing, say, 100 peptide possibilities, would you suggest any thumb rules to go after a specific epitope based on whatever you list the information? Meaning, if there are 100 epitopes, right, would there be a rank or the number that you should be looking, like the top hits?
So what you export is going to be a snapshot of what's on the details page, right? And kind of referring back to the earlier discussion we had, the default rank is by how much it's been studied. And I assume that would be the order that it would export in as well. But I think that really is a subjective question. It comes back to your preferences, right? We felt that ranking them by frequency of study, the frequency of occurrence was a nice default way to show it. But I think beyond that, it becomes sort of your decision. I don't know if anyone in the IDB team has any other input, weigh in on that.
Okay. So the second follow-up question is,
when
you make a search like that, is there any possibility to save something like a cart, in case if you have to come back, can I go and revisit that without having to go back from this scratch?
Very good question. So the question was, if you run a query, especially if it's a complex one, is there any way to save that kind of as you would in your shopping cart and come back to it later, and so that you don't have to go through the whole process again? So the short answer is, at the moment, there are no saved queries on the database. This is something we've explored. Because I'm not familiar with where it stands, I'm going to turn it over to maybe Randy or someone on the team can give us better.
You're not the first person to ask that. So
yeah,
we
get
have
that
it
a lot.
on our list of frequently requested features that we have prioritized.
Yeah, we really very much appreciate that idea. I think that this reinforces that idea that we would like. So I think the alternative at the moment is to export and have that saved locally.
As a feedback, you mentioned about tetramer.
Yes.
People use different varieties of reagents. We use dextramers. So people may left out from that type of information. Maybe you may have to use some alternative phrases to capture in a big picture. Like a flow.
That's a good point. Yeah, the comment was with respect to tetramers, there are often different versions or different reagents used, dextramers, etc. At the moment, we don't distinguish that because if you've noticed, it's sort of an umbrella term, tetramer, I believe, /dextramer, something to that effect. But it's a good suggestion that we should maybe differentiate those. At the moment, one would need to go and visit the publication to get specific details. It might be captured in the comments. But yeah, good comment. Anything you wanted to add to that, Randy?
The last point is, you mentioned about the disease diabetes in one of the queries, I think the last query. What if the disease is not listed? Can we put our own disease, like the phrase, what we will be looking for, or what if the specific disease is not listed? Say.
Okay, so the data that are in the IEDB define essentially what the search parameter results will give you. So if there is no data for a particular disease, it means there's no publication describing it. I think the good option would be, as Randy suggested, try a synonym, because it could be that oftentimes, especially when it comes to free text searches, you will have a false negative because the hit isn't coming up, but it could be something as subtle as a hyphen or some punctuation difference that causes that search result to come up. So exploring the tree. But I think that's, unfortunately, we're kind of like limited by what's in the literature in that respect. Thank you for the questions. Others, please? Yeah.
I have a question about the epitope searching feature. You were saying you can could put in your own sequence, whether it's a peptide or a full-length protein sequence, and then you would put substring as your, I guess, as your search type. I also see there's exact match, blast, all the way down to 70%. When I do the 70%, I get less hits than when I hit the substring. Is there a reason why substring has a broader search? I don't know. Is that normal, or I don't know?
Well, substring, and correct me if I'm wrong, anyone on IDP, substring is always looking for exact matches within the string.
Okay, so I'm just a little bit confused why, if I enter a protein sequence in there and I hit substring, I get, let's say, 71 results, and then when I hit a blast at 70%, I get like 50 results.
You get fewer when you blast. Go ahead, Björn.
So the reason for that is the 70% is calculated based on the length of the whole input sequence. So if you have a full protein in there, that won't work, essentially.
But if you were to
and
search
then essentially,
peptide,
if you start with the protein, I think I would always recommend going with the antigen rather than putting a sequence in and doing substring.
yeah, the antigen in the finder, it would be a good place to start. This is, in the maybe rare case that you have a specific sequence that you want to constrain it. Jay, please.
So, are there any similar databases like IEDB? In our case, we were back and forth going to this database, and we came across other databases, then we got confused. So, in relation to other databases that are out there publicly, how unique your database is so that availability is enhanced?
Yes, so our database is that the contextual part that I mentioned is the novelty of it. There are other databases, but the level or the breadth and amount of data is nowhere near. So, of course, we're going to toot our own horns. Bjorn, go ahead.
Yeah, so I mean, one example for another database is the Los Alamos HIV database. So there's a number of other specialized databases. There's also a food allergen epitope database. There's one specifically talking about only 3D structures of allergens, and so there's a number of specialized things. So that's essentially what, many of them are actually to some extent interconnected with the IDB, and then they have something that is specific for their purpose. But as Nima was saying, I think and then there's like this MHC ligand databases, and so if you have a specific interest, it is worthwhile looking for other things as well. But the whole scope is only in the IDB.
Please.
When you are looking at the HLA data in a specific population, is there any other resource to look, like geographically, which are the HLA more representative in that particular population?
Yes, so the question was, when you're looking at the HLA data, is there another resource that can help you correlate that data with geography? Answer is yes. I think I'm going to need Bjorn's help on this for the referral, but or Randy. So we do actually have the link there, right, on the HLA link. So there are ontologies. There is the HLA database, which gives the correlation of frequencies with populations. What's the specific resource, please, Mir?
Allelefrequencies.net is what we are.
Allele frequencies.
In the afternoon, actually, I'm going to show you some examples in how to use a tool that we specifically designed to project population coverage of different epitopes in different ethnicities. So we should revisit after that.
Please.
I wonder if you use other databases for the initial search for antigens or epitopes. What happens if you use no PubMed, Google Scholar or Scopus? Are you missing epitopes or antigens?
So the question was, if you use a different resource such as Google Scholar, do I understand, are you asking whether if we were to query those resources, would we find additional information? So we do, I believe, do some kind of QC level periodically and double check what we've retrieved against those other outside resources, such as Google Scholar. And there are times, in fact, that we will encounter things that we do not capture with our PubMed query. Of course, as I mentioned before, we also rely upon input from public, but we found that the PubMed query, especially because we refined this query over time, is a very high fidelity result. We get a high level of return. I don't know the metrics off the top of my head, but we're confident in it that it returns the vast majority of the relevant literature. Alex would like to.
Maybe some insight into that on a couple of levels. Yes, as Nima said, we do use Google Scholar, and specifically, for example, to make sure we capture all the tetramer data. So we have some checkpoint there. The other reason why we feel relatively confident that we capture all the data or the vast majority of the data goes back to the meta-analysis that were mentioned earlier. So in a number of instances, we have gathered experts in something, in tuberculosis or in flaviviruses and whatever, and then asked, these are basically the experts in that field, and then we asked them to double check if we captured the totality of the literature. And of course, the first thing they do is making sure we capture all their data. We know that. But very rarely they came up with things that we had missed and for which we're grateful, and obviously if they pointed out. So we have also a community double check for most specific areas. So we try to cover that by that. Yeah.
So a comment then is, do you reject the information, like based on your criteria? If some information is missing, would you reject some of those epitopes from including in your database, or would you capture anything that is published in the literature?
We do have very minimal criteria. They're not subjective criteria. They're more, as you mentioned, is there basic level of detail for us to enter the epitope. If that's not in it, we will not reject it out of hand. We will make an effort to contact the author to get that information. And in certain cases where we cannot get that, basically it won't be put in the database.
So maybe some examples of that might be if someone a paper does not provide us the structure of the epitope, and we contact the author and say, can you give us the structure? If that is not provided, then we can't. Or in other instances, it's someone will say, we used core, the epitope of core 23 to 35 of hepatitis B. I don't know what hepatitis B sequence you're talking about. So again, unless the author can provide a specific sequence to say residues 23 to 45, depends when you start counting, what isolate you have. So those are instances in which we cannot curate that information because it doesn't have.
And we encounter that quite often. There are, especially with the larger scale, epitope discovery studies and scans, microarrays, these sorts of things. Oftentimes what we will encounter is we made the entire overlapping 12-mer, profile of this protein, and unless we have a specific sequence to, reproduce those overlapping peptides, we're, as Alex mentioned, we don't assume, no matter how specific the description is, until it can be tied to a specific sequence, we will forego assuming that this is good enough. Even with really common, someone might look at you and say, "You really need me to say the sequence? This is that flu 58 to 66, the one everyone uses." But there are cases where, except for that one time where we mutated this residue in this study for this reason, and we don't want to falsify, or unknowingly present false information by assuming. So we often now will get that negative data out of the supplemental information, which is really a positive thing. But I would say our success rate is fairly high with contacting authors, in the spirit of them wanting to disseminate the information. The manuscript may not always require all the details for publication. We'll ask them for it and they'll forward it to us, and then it'll be updated, and you'll see that there won't be a table or a figure reference. It'll say author provided information.
So,
question back here. More. Go ahead.
So I'm not a frequent IDB user. I'm more like on the other side developing things. But when I use the IDB, I often encounter an issue of two things. One, I would like to have only data where the HLA allele is fully annotated, fully typed, right? And I cannot find a way to do that. Maybe I'm just ignorant. I didn't find the right query option. I did to download the whole thing. And then afterwards, I will filter the data myself to only include the ones where the MHC allele annotation is fully, AO2:01, right? Can you do that in the IDB? I didn't find the option to do that.
Do you want to take that? Yes.
Okay. So I think that's something you should put on your to-do list with high priority, because that would be very useful because moving forward, it's very important to know that. Historically, maybe it wasn't so important. AO2 was enough, but now moving forward, to fully appreciate your data, you want to know the HLA context exactly.
So the question was, the desire, actually the comment was a desire to have the ability to filter for the fully annotated allele. So for example, the certain degree of resolution, etc., right? That's what you're interested in.
And my other comment, I guess mostly also more into the team than to the users, is quality, right? So when you curate so many data points, something goes wrong once in a while, not because the measurements were wrong, but the curation process went wrong. You annotate something wrongly from copying from the paper into the database. And I don't think we're actually running such. You can run a simple check on outliers, right? And then flag them and go back and have the curators reiterate the curation.
So the question was, there's a reasonable expectation that with such high volume of data, there were going to be human error, right? And how do you check for this? So we do, in fact, run automated checks to the degree possible to identify those outliers. So the ontology in part affords that. For example, there is field A says X, field B says Y. Y is not possible when field A is X. Identify those and spit them out, and then we will go back and manually recurate those, correct them, identify other errors that spawn off of those. And so Randy actually is spearheading this kind of periodic recuration effort to do QC. She could speak more to that, but it's a great point that we periodically QC in those ways, for example.
I just want to add that we have used what you suggested where we look for outliers. We've taken binding for assays that measure binding constants. We've looked at the range of values that were entered as the quantitative value for that binding constant. and manually reviewed them and made sure there weren't errors in the outliers. But sometimes authors publish outliers, and so we take those. But I mean, we're always looking for ways to make the data more accurate, and our only main limitation is time and availability of resources. But we're always constantly trying to be vigilant and do a better job. And also, if people find errors, we want them to tell us. We want to know if something looks wrong, even if it's just a suspicion. If we have time, we'll definitely get to it and put it on our list of things to do. So I think it grows every year, and we knock some off every year.
In some infectious diseases, there is a good amount of molecular mimicry with some proteins. Is there some way to cross-examine autoimmunity and infectious disease in this database?
Yes, so good question. The question was, is there any way to look at cross-reactivity, for example, in the context of autoimmunity? There are many ways to do that in the database. One way would be, for example, when you search for the assay information, you could define like a heterologous reactivity as the antigen. So instead of it being against the source organism in the antigen field, you might look for a related organism. So they were infected with Plasmodium falciparum, but the response was to an epitope from Vivax or to the entire organism. So we do capture these cross-reactivities as long as they're epitope specific. So the very common scenario is that an antibody that is or a T cell that is shown to be specific for a particular epitope in one context also cross-reacts to an epitope in another context or to a self-antigen. And so very often if you run a straight autoimmunity query, you're going to see that you're going to get a collection of epitopes from not just self-antigens, but there may also be some connection to some infectious disease. So you can start broad. I believe Randy mentioned that earlier. We can show you ways hands-on to develop those queries to look for the cross-reactivity as well. Thanks for waiting.
Yeah, no problem. So my question was regarding the queries that you can make in the Assay tab, where you can, for example, select mass spectrometry. So I was wondering, how do you annotate those results from mass spectrometry, given that when you make an experiment of mass spectrometry, all the peptides derived from a cell line can be mapped to different HLA alleles. So are you providing all of them, or you don't annotate that, or?
So this would be, the question was, how do we annotate the alleles in assays that we capture as mass spec information? So this would be in the context of ligand elution. And so in those cases, we again follow the information in the manuscript, which will usually, hopefully, clearly state that we eluded from this allele, and this is how we know that, either because it was purified or this was a specific type of cell line. So the allele that we attribute it to will be the allele that it's attributed to in the paper. And beyond that, basically, it's sort of caveat emptor, but we do make a concerted effort to make it reflect.
You specify all the alleles, or you just, for example, if it was purified with class 2 DR antibody, then you just put DR?
We would just put that level of detail,
okay.
The class. So we will not, assume any allele specificity in those cases. The MHC types present. Go ahead. She wants more elution, but.
Yeah, so if the authors alluded using a vague MHC like that, like DR or class 2 sometimes or class 1, if they tell us the MHC is present on the antigen processing cell or in the donors that they alluded out of, we put that in a different field called the MHC types present. So it means we don't know if they eluded it from these MHC, but we know they were present in the context of that assay.
Yeah, there's another field apart from the restriction field that Randy's referring to that one can see a list of alleles that are present on the cells being studied. In those cases, you would see multiple alleles, right? But it's not.
I would use that information.
Right. In that case, the peptide is not being attributed directly. It's just, there's maybe a loose correlation there. Thank you. Are you hungry?
Sorry.
Jay, please.
I saw one of the slides where you had shown the species coverage. Is that everything, or are you expanding, like other domestic animal species? The species coverage, you talked about rodents, humans, non-human.
That was not exhaustive what's there. So the species coverage, I think we have in excess of maybe, if you go down to subtypes and strains, it's huge, but there are any species we are agnostic when it comes to species. So as long as it meets the minimal criteria, you will see anything in there.
Okay, one query difficulty. When we were searching in the database, for example, if you ask for MHC class 1 epitope by putting a specific allele, it will give different possibilities, like starting from, say, 10 amino acids, 9, 8, and so on and so forth. Based on what we have learned, at least speaking for myself, I don't think for every epitope there is information available. Why so many possibilities are given? Say maybe 8 amino acids and more, where I expect, based on what I have learned, where the information is available, that epitope makes sense, but why other possibilities are also spitting out in the spreadsheet? Does it make sense, what I'm saying?
I think it would be helpful to see the specific query in that example.
Okay.
Unless anybody in the IDB team can weigh in, I think it would be helpful to see, to give you some guidance on that in that case. Yeah. Right. Please.
I wonder if the prediction of the synthesis or specific epitope is possible to put in the database, because let me tell you something. Using your database, we selected 2 epitopes that are coming from the genome of these organisms. And we decided, because of the antigenicity, to synthesize the peptides. One of them works very well, and the other, nobody knows what happened.
So these were queried epitopes or it's predicted? So I think that the question borders on prediction in a sense, and I will defer to later. But if you tested peptides and your experimental results varied from what was reported in the database, that's quite possible. I think that's a likely thing, because we're just reflecting one experimental's results.
So, help me understand a little bit more. You said one worked very well and the other, nobody knows what happened. So it means that the peptide could not be synthesized?
Yes.
Okay, so that is not uncommon that, sometimes you will ask for a peptide synthesis and there are mistakes in the synthesis, or the peptide is difficult to make. For example, if you have something that has a stretch of 4 leucines and 3 cysteines and a tryptophan, good luck with that. But that is not a database issue. That is a peptide chemistry issue. So obviously, if something can't be made, it can't be tested. It still could be predicted, but